তাই হোক।
আপনি কোনো আছেন কোনো উইকেট অনেক দিন কত কিছু বড় বড় কিছু আছে সায়েন্স কালচার জার্নাল বিজ্ঞান পরিষদ বিজ্ঞান পরিষদের সভাপতি বিজ্ঞানের সভাপতি তাতে জিএম হবে।
I am pleased to invite Professor N D Paria on the stage, please. Professor sir, please. He will be initiating a talk on biodiversity and conservation and overview. Professor N D Paria, professor and former head of the Department of Botany, University of Calcutta, and former Vice Chancellor, Vidyasagar University, has nearly 38 years of professional experience. Taxonomy, biosystematics, biodiversity, and conservation ecology are just to name a few of his wide field of teaching and research interests. He is also the recipient of many prestigious awards in recognition of his work in taxonomy. He is the fellow of many organizations and is the life member of many others, both in India and abroad. Moreover, he held and is still holding several diversified prestigious positions. He has visited abroad more than a dozen times, and his academic career, this includes his postdoctoral work in the Department of Silviculture in Finland. He has conducted 12 research projects sponsored by different funding agencies and Government of India. Professor Paria has published 80 papers in national and international journals and has published 7 books on taxonomy and related fields. Now I request Swami Suparnanandji Maharaj, our Secretary Maharaj, to kindly honor our chief, our guest speaker, Professor N D Paria, and hand over the life of Swami Vivekananda by his Eastern and Western Disciples, Volume 1 and 2, and a book titled Science and Religion. This is as a token of appreciation and thanks for sharing his valuable knowledge and time with us. We thank Swamiji for being present in person to encourage our efforts. Before we start today's proceedings, some announcements. The initiation of discussion would be over by 7:15 p.m., followed by participation by the audience by way of questions and comments, either in Bengali or in English. Our next lecture in this series will be on 30th September 2013 by Dr. Shivananda, and the subject is Development of Solar Power Tree, a New Way to Future Energy Solution by harnessing bigger solar power consuming minimum land area. Now I invite Professor Paria to kindly initiate discussions.
Projection is चले আসবে projection और हम projection की आप भी projection laptop তাহলে laptop আমার এখানে আছে তাহলে আমি এখানে কি করব ঠিক আছে ফরওয়ার্ড ব্যাক দিও ফরওয়ার্ড দিও ঠাকুর শ্রী রামকৃষ্ণ দেব, মা শারদা দেবী, বীর সন্ন্যাসী বিবেকানন্দজি এদের প্রতি প্রণাম জানিয়ে আমি আমার আজকে বক্তব্য শুরু করছি। রেসপেক্টেড স্বামীজি, ডক্টর লাহিড়ী, ডক্টর কাম্পাত, my teacher, Professor Dotto, sitting in front of this room, and other distinguished guests, ladies and gentlemen. Indeed, it's a great opportunity for me, and I am fortunate for that. I have been invited here to deliver a talk before this august gallery, and I am going to speak on the topic biodiversity and conservation and overview. Many of you know that this is a vast topic. This is a vast topic and we cannot do justice within a single lecture. And it is a technical topic also, but I shall try to make it generalized with the help of some global, national and regional instances for the understanding of what the biodiversity and conservation are. So from that point of view, let us see the definition of biodiversity. First of all, I like to tell you that I am going to project here two definitions only. But regarding biodiversity, there are 14 definitions collected by some authors. Naturally, each definition has got different angles of approach. But here, the diversity, what we mean, that is the variability among living organisms from all sources, including terrestrial, marine and other aquatic ecosystems, and the ecological complexes of which they are part. This includes diversity within species, between species, and of ecosystem. United Nations Environment Programme, they accepted this definition. The greater acceptance of the definition is the second one, that is the totality of genes, species and ecosystem in a region is the biodiversity of that place. Actually, by this definition, the exact situation may not be so clear. I will project some slides afterwards, and you will find therefrom what the definition leads to. Now you see, as we are going to talk on the or I am going to speak on the topic biodiversity, we must remember the first Earth Summit held in Rio de Janeiro, Brazil in the year 1992. The official title was United Nations Conference on Environment and Development. In this conference, that time 178 countries participated, and afterwards till 4th June 1993, 93 parties and 168 signatory countries was there in that convention. There, 5 major documents were signed by the signatory parties, Here you can find the Convention on Biodiversity was one of them. I am not going to Rio Declaration, Convention on Climate Change, Statement on First Principles, and Agenda 21, as because our topic is today Convention on Biodiversity. This Convention on Biodiversity has got three objectives: protecting biological diversity, sustainable use of biodiversity, sharing the benefits of new products made with wild and domestic species. This is very important because regarding patent and other things, this particular objective should be maintained. But before 1992, there was no such provision. The signatory parties will follow this rule. All three they will maintain, particularly this one. You will find from some examples that what happened before 1992. You see, I will come to that part later, but the totality of genes, species and ecosystem, as we say, that is, you can now consider biodiversity as a whole, taking three together. Here you can find genetically one individual may not be, they may not be genetically similar, they look different because the same human we can find here, but they look different. This is because of genetic diversity. Very simply I am talking about this. Then species diversity, here you can find, I will show it in the next slide, higher plants, fungi, protozoa, algae, bacteria, virus, insects, other animals like invertebrates, vertebrates and so on. So, and this is the comparative chart. You will find the respective number against each category in the next slide. Then ecosystem diversity, you know that this is one, say, rainforest, here one landscape in the high altitude area. There are different types of plants, animals and microbes. They all interact together in the different ecological niches, and their collective response lead to the ecosystem diversity. Now this is the species range. Earlier I told you in the previous slide, this is the estimated number which are yet to be identified, and these are already identified different groups: virus, bacteria, fungi, protozoa, algae, flowering plants, vertebrates, roundworms, mollusks, crustaceans, spiders, mites. Popularly I can tell you that this we call Banged Chata, this is bacteria, you know, algae, salva, this is flowering plants, vertebrates, melgondi, then roundworms, creamy, shamu, then chingri, arsola, etc., chingri, makorsa, etc. Insects, large number of groups are there. Now you see the species diversity, what it is. Different types of species are there. They have got different adaptation, they have got different ecological requirement. Then we can come to the point. Why is biodiversity important? Why we should learn this? Why we should follow this? It has got ethical, historical, cultural value, ecosystem value, economic value, evolutionary value, food value. For about 30,000 or more plant species have edible parts. Then medicinal value. You will find, as we have got a large number of values of biodiversity product or biodiversity itself, there are different parties, companies, they go for biopiracy. What happened? Here you see, biopiracy, that is the illegal appropriation or exploitation of genetic and biochemical resources. Why it happens? Because the country, they lack proper code of conduct, lack of legislation, lack of national capacity to handle biodiversity utilization. Naturally, the biopiracy comes to the point. Then you see, it's a musk melon. It's a, we call fruity type of fruit. This musk melon, actually in California, they have got a multi-billion-dollar industry for this musk melon fruit. It is the raw material. In 1 year in California, there was an outbreak of fungal disease. The California government tried to control that fungal disease by every means, but they failed. They had the information that the wild gene for this particular musk melon is available in India, So they imported that wild gene of this particular plant from India. The fungal disease was checked, that was stopped, and out of that the Californian government earned a lot, and they can continue their multi-billion-dollar industry of muskmelon. India received not a single penny out of that. Then you see, this is very common plant, we know, we call it Noyantara. It grows in different tropical parts of the world. Particularly this plant is the native of Madagascar. This plant causes two important drugs. We call it vincristine and vinblastine. Now these two drugs are wonderful ingredient for the treatment of childhood leukemia. This childhood leukemia is controlled with the drug produced from this plant, and the EMA, the American giant pharmaceutical company, they patented the drug vincristine and vinblastine. Poor Madagascar received not a single penny out of that. Then you see one more thing, neem. We know our neem case. We are using the active principle as biopesticide from time immemorial. But the WR Grace, one again another giant company of America, they patented the active principle which is meant for insect repellent. So these are some of the examples what happened in the long past, at least before 1992. They used the biodiversity product material without giving any rational sharing to the country of origin of that plant. Now we come to bioprospecting. With this particular projection, you can find that in the treasures of plant kingdom, what we have got. We have got so many plants for use in medicine, for use in cosmetics, for use in agriculture, and so on. We have got large number of plants for our utilization. Now, for bioprospecting purpose, I like to project before you few slides only. One we call Stevia ribordiana. Now, it has got one active principle called stevioside. Just see what are the uses. Used in preparation of soft drinks, confectionaries, and bakery products. Induces pancreas to produce more insulin. Regulates blood pressure. Prevents hypertension. Prevents tooth decay. Useful in the treatment of skin disorders. Used as herbal medicine. This particular plant has come to our plains of West Bengal, say for last within 8 to 10 years. Now people, they are having this in cultivation, and they are giving supply to the pharmaceutical companies. Now here another is Taxus brevifolia or Taxus wallichiana. Taxus wallichiana grows in Indian high altitude area, particularly in the Himalayan belt. In this plant, Taxus has got one compound, we call it Taxol. This is a highly derivatized dicarotenoid compound. Now it is used in ovarian cancer. Now you see, this is the plant, and it has got fruit. The fruit has got red aril. The specific drug is obtained from the bark. I am coming to this point later. One thing you see, in 1 year there was a survey in USA, and out of that survey it was learned that about 21,000 females were suffering from ovarian cancer, and out of that 21,000, 12,000 would die every year. This was the statistics of that survey. Now the Taxol, it is a wonderful drug for the treatment of ovarian cancer. Problem is that if we like to treat one cancer patient suffering from ovarian cancer, we shall have to use the plant numbering from 3 to 12 trees. Now we shall use the trees means that compound Taxol, it is obtained in the bark portion. In the bark portion, naturally we shall have to take out the bark from the tree. For the treatment of a single patient, we shall have to kill 3 to 12 Taxus trees. Now, if it is so, once the time will come when the Taxus population will be wiped out from the face of the place. So the scientists, they try to search the active alternative source of Taxol. They try to search, thanks to science, thanks to God, that they could have it. That is one fungus, Taxomyces andriani. This particular fungus, it grows under the bark. Now, this fungus was isolated and cultured in semi-synthetic liquid medium. Now, this fungus could produce taxol. It will be the task of the biotechnology to enhance the production of taxol or to standardize the quality of taxol. So, that is the alternative source of taxol, which is a kind of fungus only. It can produce taxol while it is residing under the bark. Now you see, mamala plant. For HIV, for AIDS, we are afraid. Now you see, this plant, wonderful plant. This is in a flower. The plant is Malanthus nutans. It grows in the Pacific island, that is Samoa, Vanuatu, New Caledonia, Fiji, northeastern Queensland, north-south Wales. Only these are the restricted distribution of this plant. Now, antiviral extract from the bark of this tree can be taken, and that is used by Samoan islander to treat hepatitis. But its primary ingredients or chemical name, prostrating, has shown properties as an anti-HIV AIDS drug. This is in search of further betterment by analysis. Now you see, here is another plant. We call it Thaumatococcus danieli. The plant grows in the coastal belt of South Africa. It has got one protein. It is thaumatin. It's a sweet tasting protein. And at the same time, you will be surprised to know that this protein is 2000 times sweeter than sucrose, than sugar. 2000 times sweeter and low calorific value. So this particular thaumatin, it is approved as food additive throughout the entire Europe and in many other countries, used by food and confectionery industry for substituting synthetic sweeteners. Genetically, I am coming to this part in the next slide. Now this is the entire plant, this is the fruit. The fruit has got a red aril, and inside this fruit we can find this seed, and seed has got thaumatin protein, and this is the ribbon diagram structure. Now how thaumatin, that sweet tasting protein, is produced? That can be produced, you see, in this plant we can find 5 forms of genes, 1, 2, 3, B and C. Now gene 2 for thaumatin 2 isolated and cloned, genetically modified Escherichia coli and yeast with thaumatin 2 gene utilized for production of thaumatin and used in various fermentation products. As it has got 2000 times sweetness, naturally it will be desired very widely for fermentation industry. Now that sorbitol, we are familiar that those who have got diabetes, the doctors never prescribe them sugar-based medicine. It will be sorbitol-based. The natural sorbitol, it is obtained from this plant, that is Sorbus ocuparia, one of the commonest low-calorie sweetener used by weight-conscious people. Now you see, Syncephalum dulcifica, it is miracle fruit or miracle berry. This is not medicinal plant, but it has got otherwise very specific value. It is a kind of curio from treasures of plant science. You see the availability is in West Africa, Ghana, then present distribution West Africa, United States, Japan. Here we get miraculin. It's a glycoprotein. That miraculin acts as a sweetness inducer when it comes in contact with acids, causing bitter and sour foods to taste sweet temporarily. This effect usually lasts between 30 minutes and 2 hours. Now, for these particular characteristics, people in foreign countries, they hold miracle berry party. They invite their friends and neighbors, and they provide them fruits or miracle berry tablets or miracle berry granules. They consume it. After that, they provide them, supply them sour or bitter food, and they will take it as very sweet. And this is caused by glycoprotein. In fact, the miracle berry is not at all sweeter. Only thing is that it has got that active principle glycoprotein that causes or induces the sour fruit, bitter food as sweeter one. Now this is the actual fruit, this is granule, and this is tablet, miracle big. Now you see, this is another wonderful discovery or invention, what you may call it, both from plant science and animal science. You see, the name is Cordyceps sinensis. This is one moth, Himalayan moth, and this is a fungus. Now, what happens, you see, this Himalayan moth lays eggs, and from the eggs, larvae are produced. In the meantime, this fungus, they grow and mature. At one time, they reproduce through their spores, and how it is released? At maturity, this plant will release the spores in the form of powder that will settle on the soil surface. The larva from this particular moth, they consume the spores. Ultimately, they die, those larvae. And after their death, from the larvae, the fungus will come out. Now in the next slide, it will be clear, you see. Just see, this is the larva of the plant, and that fungus has died up. And this is the collection of larvae with fungus, and this is the storage. Now one thing you see, it is formed by the parasitism of sparingly branched fungus, such and such is the high weight, diameter. Now this one you see, a plant cum animal life as described in the Chinese pharmacopoeia. The Chinese name refers to the notion that it is a herb, that means a plant in summer, and a worm in winter. The Tibetan name Jorosogumbo also carries the same meaning. Now, this particular fungus, it is used in chronic nephropathy, erythema, emission, neurasthenic, rheumatoid arthritis, coronary heart disease, disease related to blood vessels of the brain, strengthening the immune system of the body. All prime elements of the body of a human body that can be cured by this particular fungus, we call it caterpillar fungus, and produced out of the moth also. Now the whole plant is taken with milk or honey as an aphrodisiac and tonic by Bhutia and Lepcha tribe of Sikkim. Here you can find that the people, they are collecting from the hills of the Sikkim. In India it is distributed in Arunachal Pradesh, Uttarakhand and Sikkim. In Sikkim it is found in between 3,800 to 5,000 meters in Chopta Valley, Muguthang Valley, Zume, Samdang, Jongli, Jodang and Kalapatthar in North District. This is the market. You can find the symbol here. That means that particular fungus will be available in this market. This will lead you inside. Now this is the storage. Now you see, what about the export situation? 200 to 300 kg per year. It is raised or harvested from the field, and the market value is 10,000 to 15,000 per kg. Rupees 10,000 to 15,000 per kg. And nearly 60 to 80 kg per year is raised from Sikkim, but that is in illegal way, not sponsored by the government. Now benefit sharing. The third point of objective of the biodiversity, I told you, one was benefit sharing. Here you see what it is. Now a classical example of benefit sharing, here you see Tropical Botany Garden and Research Institute in Kerala. They developed this term. And the interesting point is that this is the plant. This plant has got a small berry-like fruit. The Kani tribal people, they reside in the hilly area in that Kerala zone, and they popularly call this plant and Aruggopacha. Aruggopacha, this berry fruit of